Good morning. I'm Dr. Chen Wan. I'm the Director of Research Innovation, Health and Genome BC. It's my pleasure to welcome you to today's final session, Beyond Borders, Engineering Solutions in Combating Complex and Common Diseases. In this session, we will continue to explore groundbreaking developments in gene, cell, and tissue engineering. We will discuss advances beyond the DNA level that offer new therapeutic options for a variety of medical conditions, focusing on genome regulation, genetic tooling, and bioprinted tissues. Our discussion will also cover vital ethical considerations as we advance in our fight against the diseases. I'm joined by four distinguished panelists for brief presentations and a detailed discussion, concluding with an interactive Q&A session. Please feel free to submit your questions at any time using the Q&A panel on your screen. I'm now pleased to introduce my panelists, and I encourage you to read their full bio from our event portal. Dr. Carl DeBoer, Assistant Professor at the School of Biomedical Engineering, University of British Columbia. Carl specializes in developing genomic and computational tools to explore genome regulation. He will share his insights and passions shortly. Dr. Blaise Cesar, Vice President and Head of Research at therapeutics. Blaise holds a PhD in immunology and has extensive experience in cellular and genome engineering. Since 2021, she has been leading the development of IP genome editing technologies at therapeutics. Blaise will discuss her groundbreaking work following Carl's presentation. Dr. Sam Bosworth, Chief Scientific Officer at Aspect Biosystem and co-founder of the company. Sam spearheads the development of bioprinted tissues aimed at treating incurable diseases. In 2023, Aspect secured a historical bioprinting partnership with Novo Nordisk. We look forward to Sam's insights. Dr. Alice Varanin, Executive Director of Clinical Ethics and Spiritual Care Services at the Provincial Health Service Authority and a Clinical Assistant Professor at UBC. Alice brings a rich background in genetics and ethics with 3 master's degrees and a PhD. Alice will address the ethical considerations of translating these technologies into treatments. With that, I will hand it over to Carl to begin our session.
Thanks for the introduction, Chen. Yeah, so I'm a professor at UBC, and my research is trying to solve this kind of problem of how genetics underpins a lot of common complex inherited diseases. So a lot of genetic studies have been done for many different common complex diseases like autoimmunity and heart disease and schizophrenia. So these tend to be diseases that run in families, but there's not a single gene involved. And what the studies have shown is that a lot of the genetic variants that are implicated in disease, they're not actually occurring in the genes themselves. They're occurring in the regions that are nearby the genes and are thought to control when and where those genes are turned on. But the problem is we really have a poor understanding of the regulatory code that the cells are using. So we can't tell you, we might be able to identify the genetic variant, but we can't tell you what gene is misregulated in what cell type or how that connects to the disease. Could you change the next slide, please? Yeah, so a lot of what my lab's research is aiming towards is to try to understand how these genetic lesions that are causing disease, figure out how they work so that we can develop these personalized treatments to kind of rectify the effect they're having in the cell type. And because we really don't understand how the variants are contributing to disease, a lot of what my current research is focused on is developing the tools needed to generate the kind of big genomic data that will allow us to plug it into machine learning models that can actually predict how these genetic variants uh, affect disease. And actually, we're doing all sorts of research also involving drug development too, for the very few cases where we think we know enough about how the variant works that we can actually design drugs to treat them. Thanks.
Thank you, Carl, for the very really good overview. And we're going to dive into some of the tools and approaches you are researching and developing a little bit later in our discussion part. So with that, now we will hear from Glenn.
Hi, thank you for the introduction as well. So like Chen said, I'm the head of research for an exciting startup biotech company that's actually based in two places. We're about a 50% split between Seattle, Washington, and Durham, North Carolina. This company was started about 4 years ago, right at the beginning. The for our company comes from the laboratories of Dr. Charles Gershbach at Duke University and Dr. Fyodor Urnov from the University of California at Berkeley. And we're focused on developing tools to enable epigenetic editing for clinical treatment of diseases. So next slide. So we know that most diseases, like Carl had said, are not necessarily due to changes in the genetic code, but are actually due to misregulation of the patterns of gene expression or changes in the epigenetic regulation. So at Tune, our aim is to harness the power of the epigenome to dial gene expression up or down or in combination for the treatment of a whole spectrum of diseases. We all know that every cell in an organism essentially has the same DNA. So it is epigenetic mechanisms such as DNA methylation or histone modifications that really determine what genes are turned on or off in order to give us a unique cellular phenotype. So we can harness these same proteins involved in the epigenetic processes to engineer novel tools, which we can then use to alter gene expression for therapeutic applications without cutting or nicking DNA. Next slide. To be able to achieve this, we've developed this modular platform that we call the Tempo Tuning Platform. So the first element of the platform is shown on the left. And this is really a DNA binding domain, so something like a CRISPR-Cas that's been modified to have no nuclease activity or zinc finger. And this domain enables us to pinpoint any target with exquisite specificity and also allows us to control the size and the potency of the overall construct. And then to this DNA binding domain, we can fuse one or more effector domains. And at Tune, we've developed a vast library of these effectors that can impact a variety of epigenetic mechanisms. So for example, some of them can recruit transcriptional machinery for gene activation. Others can bring heterochromatin forming factors to repress gene expression. And some others can result in either DNA methylation or demethylation to achieve more stable repression or activation. So these tools can be delivered for in vivo applications using LNPs or lipid nanoparticles or AAVs, depending on the use case, or using electroporation or lipid nanoparticles for enhancing ex vivo cell therapies such as CAR or TCR products, as well as iPSC-based products. So we currently have our first product that is moving into the clinic this year using an LNP-delivered epirepressor that we have shown can be delivered in a single transient dose and actually can shut down HBV infection. And so we're very excited to share that, and we're going to have clinical data hopefully next year. Thank you.
Thank you, Bless. And indeed, groundbreaking and very interesting. I think our audience perhaps already started curious what's the difference of genome editing, which we're going to discuss again later. And so switching to Sam now.
Thank you, Jen, and thanks everybody for being here today. So I'd like to tell you a little bit about myself and Aspect Biosystems. So I'm Chief Scientific Officer here at Aspect. Aspect is a Vancouver-based biotech company that spun out from UBC quite a few years ago now. And we spun out from UBC with a unique bioprinting technology. And we're applying that therapeutically by bringing together unique combinations of biomaterials and therapeutic cells to generate bioprinted tissue therapeutics. There are many diseases in the world that are the result of loss of cells or loss of cellular function. And these tissues are designed to replace, restore, or supplement those functions that are lost in disease. So if we go to the next slide, please. Thank you. So this video really shows how we were able to marry the technologies of microfluidics and 3D printing. So this video shows one of our microfluidic printhead devices. We're able to bring together biomaterials in liquid phase, combine those with living cells on the printheads, and then cross-link those materials almost instantaneously to generate cell-containing hydrogel fibers. Those hydrogel fibers are then deposited from the nozzle of the printhead, and we're able to lay down that cell-containing fiber building block. We lay those fibers down to generate these bioprinted cell-containing tissue therapeutics, which can then be implanted into patients to, as I said, restore or replace function that's lost in disease. So next slide, please. And if you can click through all the way to the end of this slide, that would be great. Thank you. So as this is a 3D printing technology, we're able to control a lot of the macro architecture of these implantable devices. And what I mean by macro architecture is things like the distance between the fibers that are generated, the number of layers of the device, the overall fiber patterning of the device. But uniquely, we're also able to control the microarchitecture of these implantable devices. And these tissues at the microscopic level, as I said, consist of cell-containing fibers. But if we take a cross-sectional look at one of these cell-containing fibers, you can see that each fiber contains multiple shells or layers. And this is just one example of how we do this. But each of these different layers can contain a different biomaterial. And those biomaterials are optimized for a specific function. So, for example, in the core of the fiber, we have the cells that reside within a biomaterial that's specifically designed to support function. Surrounding the core, shell 1 is a cell-free biomaterial that really provides a level of immune protection. These are allogeneic implants, and the host immune system would target the cells for attack. And so we have this semi-permeable membrane that acts as a physical barrier to prevent the host immune system from accessing the therapeutic cells. And then shell 2, surrounding the outside of the fiber, is really designed to support the appropriate host tissue response to these implants. And these are all different materials that are focused on just performing a very specific job. We're not trying to find one material that can perform all of these different roles. So that's what really makes this technology and this approach unique. And then if we go on to the next slide, please. Chen, you mentioned that we recently signed a groundbreaking deal with Novo Nordisk to focus on developing therapeutics for type 1 diabetes. But there are many diseases where providing a replacement to cell function could really have a major impact on patients. And so we have various endocrine disorders that we're focusing on, as well as various metabolic disorders, particularly in the liver space, where replacing a liver function with a hepatocyte-containing tissue could really have a huge impact on patient health. So with that, I would like to finish and happy to answer questions later on.
Thank you, Sam. Indeed, truly exciting to see all those potential applications. And so last but not the least, let's hear from Alice. And it's so important to take into the ethical considerations into those discussions. Over to you, Alice.
Great. Thanks so much for having me today, and appreciate the opportunity to speak to you about some of the intersections. My clinical background is in genetics as a genetic counselor, and for a couple of decades now, I've been super interested in the intersection between genetics, genomic technology, and ethics. And when we think about ethics, I've tried to show a kind of a picture of a mountain here, trying to give us a sense of like some of those bigger picture questions we might be thinking about. I think with these types of technologies, we always need to think about, what we can do and what we should do. And so those are some of those big overarching questions. And then I think what we're also tasked with is thinking about, okay, what are the ethical questions at a societal level, at a community level, perhaps, at a national level that we should be grappling with in terms of what research we're looking into, etc. And then there's ethical issues and questions, in the application on a patient, family, population level. So those questions need to be explored and examined. Next slide, please. It's interesting. The slides are coming up super fuzzy for me, so I can't really read them. I hope you can. But some of the ethical issues that I wanted to highlight, which have come up with, that's better. Thank you. That have kind of been discussed for the last, at least the last 2-3 decades since I've been involved in this work, is things around unintended consequences. So, some of the technologies which you've just heard about, super exciting, super interesting, but what unintended consequences might there be both on an individual and societal level? What are the harms? What are the risks? What are those longer term health and environmental risks that we need to be considering? How do we really grapple with questions around human dignity and autonomy, and autonomous decision making, not just at an individual level, but at a community level? And what does informed decision making really look like? One of the big questions that I hope we'll have a chance to explore more in the discussion period relates to equity and access. When we think about who is accessing the benefits of these types of technologies, how do we decide which technologies we invest in? How does that impact equity and access? And then questions around enhancement versus treatment. And so, for example, when do we think that use of gene editing, for example, could lead to a gradual normalization of it for non-therapeutic advancements? And are there genetic inequities that could come up? Next slide, please. So I also hope we'll spend some more time talking about this, but I'm super interested in some of the new ethical considerations and concerns that technologies might be presenting us with, which might be different from some of those which we've talked about in the past. So things about relating to human health span, public health impacts, preventative medicine, regional differences and how that may play out on global equity, and then again, access to therapeutics and what that means in terms of socioeconomics, etc. Thank you.
Thank you, Alice, and thank you all for the wonderful overview of what you do and what you care about. So let's dive into our discussion part of this panel, and perhaps let's start by asking some fundamental questions to further understand the basics of the technologies and as well as the ethical concerns. So if I can start with perhaps Bless, who joined us from far. I think it's very important to understand how epigenetic editing diverge from the standard gene editing, and which is crucial to appreciate, right, its unique challenges and potentials. So I'm wondering if you can elaborate on exactly how epigenetic editing different from the standard gene editing.
Sure, happy to do that. So the bottom line is that epigenetic editing typically involves a DNA break, whether that's a double-stranded break or a single-stranded nick if you're talking about standard gene editing or base editing. And many times it also involves introducing genetic material to that break. Whereas epigenetic editing is actually just harnessing the DNA binding domains as a kind of a homing beacon to the specific point in the genome you want to go, and really is more as a shuttle. And so you're using that to bring epigenetic editors to that point in the genome to mediate different epigenetic changes, but those changes are not to the actual DNA sequence itself. It's to the epigenetic, like basically landscape. So whether you're removing or adding methylation, you're not actually changing the DNA sequence itself. So it allows you to be able to look at to modify multiple genes at a time without the risk of genomic instability and translocations and things like that. So that's the main difference.
Thank you, Blythe. Indeed super interesting. And so I also have a question for Carl in terms of, the approach and the tools you're trying to develop. In particular, you mentioned you're actually using and applying machine learning algorithms. So I'm wondering if you can maybe just give us a bit more idea around what kind of machine learning algorithms do you use to learn how DNA works?
Yep. So in general, our kinds of machine learning models usually take as input the DNA sequence and are trying to predict its regulatory activity in cells. So like whether it's it has a specific epigenetic state, as Blythe was talking about. Some of that can be determined from the DNA sequence, and gene expression of nearby genes. So that's kind of the problem they're trying to solve, predicting the activity of the sequence from the DNA sequence itself. And these are important for understanding these disease variants because basically you just put in the allele that is associated with disease and the one that's not, and you can tell by the predictions of the model what the differences are. So in general, we use two types of computer models. One of them is called a deep convolutional neural network, and this is kind of a black box style model. They don't they're very difficult to figure out exactly what's going on in the model, but they tend to work very well. And sometimes all you really care about is how well the predictions work. In other cases, we want to know more about what is actually happening on the DNA, and in those cases, we're developing a related type of model called a physics-informed neural network, where basically you just set up the model in a way where it knows about the rules of this is the way biochemistry works, except you don't know what any of the molecules involved are or how they work, and the model has to learn all of these parameters, and it learns them in a very similar way to a neural network. So we use different models for different approaches, and they both have strengths and weaknesses.
So Carl, that's great. And maybe a little follow up to that, and is so by developing, like using different models, developing those tools, approaches, I know you mainly focus on like the fundamental research, but can you also maybe speak to what kinds of cell disease models that you're working with and also potential applications?
Yeah, so we're more of on the technology development and machine learning side of things, but we work with various collaborators who are more experts in the disease areas. And part of the reason we take that approach is because our models and approaches are applicable to almost all common diseases, so we can't solve all of them and rely on the expertise of collaborators. But in general, we focus on two types of disease: autoimmunity and heart disease, in particular atrial fibrillation, which is a common arrhythmia, especially in older people, although some younger people also get it. And for those, we use things like immune cells that we can test in vitro, and stem cell derived cardiomyocytes as like a model for heart tissue.
Okay, so that's great. And maybe I can switch a little bit to Sam. And I know you talk about the implants and the really cool bioprinted tissues and the potential for them. And your implants contains allogeneic cells. And I'm wondering, like, what are your strategies to protect the therapeutic cells from host immune attack while keeping the cells alive?
Yeah, thanks, Jen. It's a good question. So yeah, obviously in autoimmune diseases like type 1 diabetes, the cells that are generating insulin are targeted by the immune system and killed off. And so going with an autologous cell approach isn't really possible. Same thing with other inherited metabolic diseases where the cells are missing specific enzymes. So we have to really use an allogeneic cell source, whether that's stem cell derived or cadaveric. Um, and really, the cells within the tissues need to be protected from the host immune system in that allogeneic setting. But we don't want to put the patients on long term systemic immune suppression uh, due to the increased risk of opportunistic infections and also the increased risk of cancer. So we need to protect those cells without giving the patients really severe immune suppression. And the strategy there is, and what I showed in the example was, we used the biomaterials to physically protect the therapeutic cells from the host immune system. So the fiber-based building blocks have multiple layers to them. In the example I gave, the therapeutic cells are within the core of the fiber, and they're surrounded by a shell of semi-permeable hydrogel. And that cell-free shell enables the free diffusion of oxygen and growth factors from the host into the therapeutic cell containing core, but it physically prevents the influx of the host immune cells into the core where the therapeutic cells reside. So we enable the diffusion of growth factors, nutrients, and the therapeutic factors in and out of the cell containing region. But those semi-permeable hydrogels physically prevent the cells of the host immune system from accessing the therapeutic cells.
Yeah, that's indeed very, very interesting, and really appreciate the detailed descriptions and explanations of the different shells and their functions. Really, really cool technology. And just back to Bless a little bit. So you talk about the difference between the epigenetic editing. You also talk about a little bit of the delivery, the potential delivery strategies you can use, like with the virus, AAVs, or lipid nanoparticles. I'm wondering, like, you kind of trying to develop the clinical products use the epigenome editing technologies. What are the biggest challenges?
I think one of the biggest challenges is really this is a new modality to go to the clinic. So a lot of people are familiar with epigenetic modifications in CRISPR-I and CRISPR-A, but these are really in research functions. So being able to go in and make epigenetic changes and show regulators that what you've done is safe has a different set of challenges than kind of standard gene editing. And so I think one of the things is just developing the assays to be able to look at, when you've done an epigenetic change, do you get the transcriptional and protein changes that you wanted, whether you're tuning a gene up or tuning it down? What other epigenetic changes have been made, if any, and are they relevant? And being able to, understand that and explain that to regulators, I think is important as we move into the clinic. It's just a different set of questions that we have than standard gene editing.
Yes, so thank you, Bless. Maybe a little bit of follow-up. I kind of imagining you perhaps have like close interactions or conversations with the regulators. And just wondering, like, if you can give us some insights around like those conversations and whether or not they are able to kind of work with you closely in terms of, either the guidance or maybe work together on what kind of data, what kind of assays are needed for the future regulatory approval.
Yeah, absolutely. So we've had great interactions with all the regulatory agencies that we are starting to work with. Different agencies have different requirements, though things are getting a little more streamlined across the world. But most of the regulatory interactions have been like partnerships. We come and we make the case for what we're looking at and why we're looking at it, why we think it's relevant, and obviously they're first and foremost looking out for the safety of the patients. So there's a lot of back and forth discussion on whether or not looking at, say, in the case of overall methylation, if you're using a methylating agent as your epi editor, what is the relevance of methylation if you see no changes in actual gene and protein expression? And so just trying to understand that and the impacts over time. I think that the regulators are quite reasonable, as well as long as you make the case for the rationale on what you're doing and why you're doing it, and making sure the patient safety comes first.
Thank you, Bless. That's really great to know. Like in our day-to-day and also our projects being funded, like if you're kind of developing new technologies, that the regulators also doesn't really have the experience in terms like, okay, what do they need to approve? So it's incredibly important to work closely and early with the regulators. So as a follow-on, so Sam, I also have a question for you in terms of regulations. So how would those bioprinted tissues be regulated? Are they being considered as medical device or biologics?
Yeah, great question, Jen. And they in our interactions with regulators, they're really considered a combination product. And so they're part biologic and part medical device. Now, the cells are really the primary mode of action. So when we talk to regulators, if you take the FDA as an example, it's the Center for Biological Evaluation and Research that is the main group that really is responsible for regulating these tissue implants. Because, sorry, there's just some background noise, I think. And so it's CBER that is responsible for driving the regulatory kind of interaction, but they would have input from the device group as well, the CDRH. So the cells are taken as being the primary kind of therapeutic mode of action, and then the device part, the biomaterials part, is almost kind of a support piece of the technology. There are definitely paths to kind of accelerate regulatory review with these combination products. There's the RMAT designation, which can at least prioritize the file from the regulator's perspective. And then there's a few other kind of approaches as well in different jurisdictions that can help accelerate the path for these types of combination products. But yeah, that's really, I think the answer to the question is it's treated as both a biologic and a device, but it's the biologic that gets most of the attention as it's the cells that are the primary mode of action.
That's great. Thank you, Sam. And so as we can see, right, that we have like a lot of exciting advancements and exciting technologies really trying to come up with those exciting medicines to really treat currently incurable diseases. And so, Alice, when we kind of look into those really advanced technologies, I know you touched on the potential novel ethical issues a little bit at the end of the presentation, but can you maybe just elaborate on that? And what are those novel considerations when we talk about those technologies that we haven't seen before in genetics and genomics?
Thanks for the question. And I think maybe I'll just start out by saying I don't think there is necessarily anything which is so new and different that we haven't thought about over the last 20, 30 years when we've been talking about some of the ethical issues in genomic technologies, and certainly questions which are coming out in the last decade or so around CRISPR technology and gene editing. Many of these are the same discussions. Where I think we've had a little bit of a shift is that I think we're coming closer to those really crunchy questions around therapy versus enhancement. I think we're coming close to those really crunchy questions around access and different countries, different people's perspectives on what is okay and what is not okay. I think we're being forced to answer crunchy questions around, okay, well, we've had some moratoriums in place, but who gets to decide? Like, who has the moral right on some of these questions? Some of the areas where I don't think we've had enough focus, which I would like to have, more discussion on, and I think that the ethics community is beginning to kind of make noises in this regard, is that what are some of the environmental considerations and impacts from these types of technologies? What does it mean if we are able to expand lifespans, by 20, 30 years? What's the impact on a global perspective on that. So I think that's one of the questions which is coming up, as well as a lot more questions related to trust in society more broadly. We've seen how trust in the healthcare system, how public health, and how trust in technology has really been pushed over the last number of years with the pandemic. And how do we really grapple with some of the fallout from that and how it's going to impact uptake and applicability? Um, the other thing I want to touch upon, and perhaps can touch upon more later, is equity. And it's interesting, Blythe, you talk about, the partnership with regulators right from the get-go, which is great, but who's asking the equity questions right from the get-go? And I think those are increasingly on a societal level things that we are having to discuss and grapple with on a meaningful level.
Yeah, so that's great. You mentioned equity because this is like the next thing I would like to ask, right? How do we actually ensure that equity is front and center, basically in those discussions when we kind of develop and deploy those really advanced technologies in treating like human diseases? So I'm wondering whether like the rest of the panel would like also to comment on that. What would be your considerations in your development?
Yeah, I could comment on this because actually I was just talking to a group about this yesterday about a lot of these genetic medicines, whether they're like CAR therapies or some of the gene editing therapies that are coming out costing hundreds of thousands or millions of dollars. They're just inaccessible to most people, and insurance companies are slow to cover them. So finding a way to make this more equitable, I think one of the challenges is the length of time and what is needed to be done to actually develop these drugs. Companies do need to be able to recoup the money that they spent, and many times it's a decade of work and a lot of failed hypotheses before you get to a drug. And so how do we balance the cost of making these drugs with the cost to the patients? And this is something that we talk about a lot. And one of the reasons we like using LNP technologies is, the whole platform was built around that for the COVID vaccine. So there's a lot more scale up manufacturing, and hopefully the costs will come down as time goes on. But I think it's a really important question.
Thank you so much, Bless. Does any of the rest of the panel would like to also comment on the equity considerations?
Yeah, I'm happy to give my thoughts from aspect perspective. I think when we signed our deal with Novo Nordisk, it was obvious right from the start that their intention is to try to make this bioprinted cell therapy as broadly available to type 1 diabetes patients as possible. So their goal is not just to provide a therapy that could hit 1% of these diabetics, but it's really to potentially replace injected insulin across the entire patient population. And so in order to do that, there are going to have to be kind of cost reductions over the course of the lifetime of these products. The stem cell derived islets are really the largest cost component for a bioprinted therapeutic. And as we scale the cell manufacturing to treat the million T1D patients in the US, then there is going to be cost savings with scale. But these are still going to be more expensive products than just the basic injected insulin for sure.
Thank you, Sam. And I'm going to incorporate a audience question here because it's really related to this equity topic here. And I think this question is addressed to Carl. So how would the fundamental researchers, if this is needed, necessary, incorporate the regulatory and ethical considerations into their project design?
Well, the short story is that it depends on the project design, but we do think about this quite a bit. There's historically been a lot of inequity in genetics research, especially over the last 30 years or so since the human genome was sequenced. There have been subsequent genetic studies that have been heavily biased towards people of European ancestry. So our genetic understanding of disease is much better for those populations, which obviously for diagnostic purposes means that if we're using these to help stratify patients into those who are getting treatment and those that are not, if we understand the disease architecture in Europeans much better than in other populations, that leads to inequity in treatment, whether or not the patients themselves can afford the treatment or whether or not society is willing to pay for it. One of the reasons we're pursuing the approach we are is because once we have the computer model that like kind of understands how the genome works, it should work almost identically in any ancestry. So that will really level the playing field. It doesn't matter now if the genetic studies have been done in that population. We have now a model that can just tell you the answer for that group as well. It's important, like a lot of our science too, we're thinking decades into the future, and it's really hard to tell the future, but it's important to try and think of all the potential ramifications of what you're doing. So we try to do that as well, but it is a challenge.
Thank you, Carl, like by acknowledging the challenge, but also kind of offer some hope. So before we get into the list of audience questions, and I do have a couple questions I would like the panel to address. So one of the questions is a little bit looking to like the future, kind of future directions and the future considerations. So Sam, like in order to get patient access, and you have to think about the manufacturing. So how and where would those bioprinted therapeutics be manufactured?
Yeah, good. Thanks for asking the question. I'll make one more comment about the patient equity piece as well, I think,
Sure.
before I talk about manufacturing. So one of the attractive features of a therapy based on a cell therapy that really reproduces or replaces a lost function in a disease like diabetes is that that same therapy could be applied across all patient populations that have that particular disease. So it's not a therapy that's designed to just treat one subset of patients. It could really treat all diabetics, and same thing goes with the other therapies that we're developing as well. So from a efficacy perspective, it's these therapies really have a high equity component. As far as manufacturing is concerned, the bioprinting technology is in itself a manufacturing technology. So we bring together the cells and the biomaterials to create and generate these tissues using our technology. And so there is an opportunity to build manufacturing capability here in Canada, and our intention is to be an anchor company here in Canada that's able to perform clinical manufacturing of these tissue therapeutics here in Canada. So that's our goal, and we're our plan is to build out that manufacturing capability. The larger scale manufacturing for commercial scale is something that is still to be determined. There's options there, potentially going through CDMOs or building out again our own larger scale commercial manufacturing. But we certainly intend to build the clinical capabilities here in Vancouver.
Thank you, Sam. And indeed, very exciting and inspiring to kind of the aspect of trying to become the anchor company here, and that's definitely what we need as Canadians and also for BC. Just one more question before we go to the Q&A session with our audience. So Bless, I'm wondering what can epigenetic editing do that other types of drug cannot do?
Yeah, so one of the great things about epigenetic editing, like I mentioned earlier, is that you can actually tackle epigenetically editing multiple pathways at once. And you can tune genes up and down a little more precisely. So many times when people are trying to overexpress a gene, they're using lentiviral or adenoviral type constructs and pushing very, very high expression, non-physiologic levels of expression of a gene. Or conversely, when you do standard gene editing, many times you're just knocking a gene out completely and forever. And with epigenetic editing, you can tune a gene down maybe halfway, depending on where you place the epigenetic editor on the DNA sequence. You can also tune the gene up or down for different durations of time, depending on the type of epigenetic editor you use. So which effector you tether to that DNA binding domain. So the modular aspect of epigenetic editors gives you a lot of flexibility on how you can alter gene expression. And the other really cool thing that epigenetic editing can do is actually direct cell differentiation. So in the case, actually, Carl and I should probably talk after this, of differentiating iPSC cells into different cell types. You can utilize epigenetic editing to transiently turn on different pathways to drive cellular differentiation down different pathways. And the nice thing about this kind of transient tuning up and down of different gene combinations is you can actually tackle these more complicated questions of cell differentiation in a way that no other technology really can do.
Yeah, this is really incredible and really cool, and I definitely hear that this precision kind of tuning kind of really needed here. So let's kind of we got a little less than 10 minutes left. Let's go to the audience questions, and I would like to direct the first question to Alice. So Alice, do you think it would be appropriate for an international body to make ethical rulings on these treatments, or should it be done on a jurisdictional level?
Thanks so much. It's a great question. You know, there's two parts of ethics which I think are helpful here to tease apart. One is in ethics, we spend a fair amount of time talking about good process and procedural justice, and the other is in ethics we talk about, okay, what principles and values should we uphold in terms of um, uh, you know uh, balancing what's right, what's important, what goals we have. I think what we're talking about here is, how do we have good process? Because ideally, we would have an international body who could come together where everyone's voice was at the table, where there was a good process to ensure those voices who are usually not heard from, have a place, that we can actually come together and agree on what, as a global society, we think is important, etc. So that's the ideal. But can we get there? Can we get there with good process, and can we make sure it's an equitable process and we have those groups who are usually underrepresented at the table? If that's not possible, how do we have jurisdictional level types of decisions and discussions? I think the jurisdictional level is super helpful in terms of thinking about, okay, well, what's the impact on us as a nation? How are we going to pay for these therapies, not pay for these therapies? What do we think is acceptable and not acceptable? However, unless we act with that global lens, we're not really getting to the heart of those questions around, okay, well, what are the environmental impacts here, which are going to impact everyone? What are the impacts here which are going to increase or decrease global disparities? So unless we have that global lens on it, we're not going to get to the core of some of these really important ethical issues.
Yeah, that's really important to hear, and should be appreciated the importance of both having the international and also the jurisdictional kind of considerations. So we got a couple questions actually for Sam from our audience. Like one of the questions is, so in device trials, there can be accelerated scarring that is immune dependent. So how do your treatments consider this going into trials?
Yeah, thanks for the question. Just to clarify, the question is about accelerated scarring as a response to the device. Is that right?
Yeah, in device trials, that can be accelerated scarring that is immune dependent.
Yeah. Yeah, exactly. No, it's a good question, and something that is definitely an area of focus for us. And so, because the cells, the therapeutic cells, are sort of embedded within the biomaterials of the tissue implant, there is no direct immune interaction with the therapeutic cell. However, um, it's a pretty common observation in the field to have a kind of a fibrotic response to biomaterials that are foreign. And so, one of our approaches is to really enhance a more positive response, an interaction between the host tissue and the biomaterial of the implant. So, if you remember in that multi layered tissue that I showed, that outer shell is a biomaterial that's selected to really enhance and optimize that host tissue response. And so, that host tissue response that we're trying to optimize can include things like encouraging vascularization around the tissue implant, reducing a innate immune response that might lead to fibrosis. And so there's a variety of biomaterial strategies that can be applied there. But we're really trying to, instead of just hiding that tissue implant from the host, we're trying to harmonize the interaction between the tissue implant and that host tissue.
Thank you, Sam. And a question for Bless. Is the epigenetic tuning done by Tune Therapeutics permanent and reversible? Or reversible? Is permanent or reversible?
So it really depends on which epi editor we're talking about. So we do have epi repressors that actually mediate methylation that is quite durable. Now, theoretically, you could use a demethylator and remove that methylation with a separate product. So it is reversible. But depending on the use case, you may not want to reverse it. Many of our other epi editors are more transient. So you may upregulate a gene for several weeks and then it slowly comes back to normal, which may be what you want for the clinical use case. So to answer the question directly, yes, it can be permanent, but yes, it also can be reversible.
Thank you. Maybe like a little bit of follow-up is like, are there any opportunities for combining what you do, which is epigenetic editing, with other treatment modalities such as gene therapy or maybe small molecule drugs?
Absolutely. So I think, one of the things that we like to think about is, cellular differentiation. And so, like I mentioned, the iPSC. So combining iPSC derived cells in something like a biomaterial, like what Sam is working on, is the kind of technology that we would want to combine, as well as many people have different technologies for different delivery mechanisms. There's a lot of AAV capsid research out there for delivering payloads to different tissues in the body. We don't necessarily focus on developing those. So those are the types of technologies that we would pair epi editing with to be able to kind of capitalize on those delivery technologies to deliver our epi editing technology to tissues maybe that aren't as accessible with standard platforms.
Thank you, Bless. I think we have a little bit more than 2 minutes left. So I would like to ask the last question from the audience, and all our panelists, please feel free to speak to it. So with like our patients getting more complex and in their ongoing care, in particular, taking into the considerations of our aging population, which is really increasing. What is your perspectives, like how those technologies that kind of targeting the more complex and common diseases would help reduce the care burden?
I think I can just have a quick kind of contribution to that question. So, what we're trying to do is develop therapies that don't just kind of support the patient to deal with their disease in a better way. We're actually trying to create a functional cure, and so whether that's for diabetes or other congenital liver disorders, it's not a drug that's trying to just improve quality of life. It's a therapy that's actually trying to provide a functional cure. So I think that's a different approach to therapeutics in general.
Thank you,
And Blythe,
Sam.
talk about PSC differentiation.
Right? Yeah, I would answer as well for kind of aging populations. When we think about how we could utilize our epigenetic editing tools, I think it really is around iPSC cellular differentiation for kind of tissue regeneration. So thinking about joint and cartilage cell regeneration or other tissues, being able to take an iPSC and direct that differentiation by activating the right combination of transcription factors, to be able to get fresh cells, off the shelf cells that can help in elderly populations. That's a kind of a long-term goal of ours. Obviously, it's more complex than single gene regulation, but it's something that we think that the technology will eventually be able to do.
Awesome. Carl and Alice, would you like to offer some final thoughts?
Sure. So just to echo what Sam said, these cures tend to be more expensive upfront, but long term much less expensive. And I think a lot of our current healthcare issues are partly resulting from the disease not being prevented in the first place. And then once the disease happens, or it gets much worse, then it's much more expensive to treat. So re-emphasizing disease prevention through potentially curative treatments or other therapies would probably end up saving the healthcare system quite a bit of money.
Thank you, Carl. Alice?
Yeah, thanks for that. I think what I would just add is that, clearly, cure is something which a lot of folks can get behind and say, okay, this is what we're looking for. But I also think we have to look at some of, what the potential consequences of that could be. Like I think about the impact of Trikafta on cystic fibrosis, and in terms of extending life here by decades for people who initially thought that they would live till their 30s or 40s, and what that's meant for them in terms of, okay, my life now looks very different than what I thought it would look like, and I've not planned for it. So I think we need to, as exciting as these technologies are, we also need to think about bigger picture consequences for society, and how we can, put in place supports, recognize psychological impacts, etc., as we move along that journey.
So thank you. And with that, I would like to conclude our panel and really appreciate our panelists for their time and invaluable insights, and also for our audience for your engagement and all the great questions. So while we couldn't address all the questions today, we look forward to continuing those vital discussions and fostering future collaborations, which I already saw happening within our panel, and that will really benefit mainly through innovative technologies and precision medicine in BC, in Canada, and beyond. So we hope you found this conversation enlightening, and I will now hand things over to Federica for some closing remarks. Thank you all for joining us.
I've successfully unmuted myself. Thank you very much, Chen. I want to really conclude this first day of a phenomenal Genome BC annual genomic forum by thanking everyone involved so far. I'd like to really thank the speakers, our keynote speaker, the panelists, the moderator, and all of you participants for the engagement and the participation. We have heard some pretty amazing and remarkable advances in genome engineering today, and what they actually offer in the field of healthcare. And what they offer is a glimpse really into our future, into what medical science really can empower for a better health outcome for all of us. Certainly, we've heard this, but we've also heard that this is not just a bunch of methodological tools or new innovative approaches coming online. Is also an important and responsible area of work. Ethical consideration must be included, and voices heard right in the beginning. This is very, very important for sure. We in this panel today, especially, we've heard about the CAR-T innovative approaches, which was great. We've learned that Canada has approved, in fact, the standard of care in British Columbia for blood cancers, and we've heard about the potential broader applications in rare diseases, in autoimmune diseases for the future as well. And we stretched it as well to understand beyond the DNA what other therapies can be done in the regulatory space and in the epigenetic space. And of course, we heard everything about the some of the tissue printing approaches and therapies as well. So I think we have a lot to reflect on, and there's definitely a lot of information we should take with us today. And I will love to welcome you again tomorrow morning for another chapter and another amazing morning where we are going to explore genome engineering for the sustainability and environmental conservation. So we're going to look at different eco innovations in this space. So for now, I thank you all again, and I will see you tomorrow. Thank you.